Good morning, everybody. How are you? Great.
Yeah.
It's time for day 3, my favorite subject, proteomics.
Mine too.
Grand. Thanks again to Diplomics for potentially funding a whole series of lessons across the country on this subject. Let us move ahead with proteomic identification. It's lovely. You could go to many bioinformatics classes offered worldwide and hear not the first word about proteomics. A lot of people assume sequencing and bioinformatics go hand in hand. But in fact, the use of sequencing data to identify proteins is a huge theme within bioinformatics, and a very large community of bioinformatic researchers around the world have been focused on problems in understanding mass spectrometry data sets for a long time now. So I'm really glad that you're going to get some exposure to how we go about identifying proteins. So let's move ahead slide here. So today we're going to start with an introduction to tandem mass spectrometry. I think rather more people have the basics of how DNA sequencing works than how protein identification works. So I'd like to try to explain the nature of the data that we're working from. We're going to talk about database search algorithms, which are the standard workhorse that pretty much everybody in the field uses. There are other ways to analyze data sets from proteomics than database search, but this is far and away the most dominant method out there. Almost any paper in proteomics will include the use of at least one database search algorithm. From there, we're going to talk about evaluating whether the identifications produced by database search are viable or not. Do we need to set the bar here or here or here in order to get a particular false discovery rate? So we'll talk about that in the context of Target-Decoy, a very popular method, or distribution fitting, another popular method that have sort of coexisted now for more than a decade. And finally, we'll talk about protein assembly. One of the things that I hope everyone takes away from this is that when we do database identification, tandem mass spectrometry experiments, what we are identifying directly are peptides. So when we give you information about which proteins are coming back, it's based on which peptides we see. So I'll explain that as we move along. So what is proteomics? There are lots of different definitions out there. Kind of the conventional one that you hear is that just as genomics studies the full set of genes available in a species, proteomics studies the full set of proteins in a species. But we can't really do this in the way that genetics does. Just as we needed to measure transcriptomics to see which genes are being expressed out of the cellular catalog, we need to do proteomics because we want to know which proteins are being expressed. And that's going to change quite a lot. In a skin cell or a neuron, you're going to have very different cohorts of proteins that get produced. So the Human Proteomics Organization, HUPO, is one of the biggest organizations in the world devoted to this topic. So they describe it by saying proteomics focuses on the identification, localization, and functional analysis of the protein makeup of the cell. The proteins present in a cell, together with their function, subcellular location, and perhaps even structure, change dramatically with the organism, and the conditions faced by their host cells, including age, checkpoint in the cell cycle, and external or internal signaling events. Just to give one very brief example, we know that lots of proteins don't really take on an active role until they are phosphorylated, at which point they take on very active roles in cells. Is it possible to tell whether or not a protein is phosphorylated based on its messenger RNA? Not at all, not at all. So unless you're able to study protein structure directly, you don't have much grip on whether or not it's been modified in a way that allows it to take on an activity. Okay, so let's move ahead then to talk about a major shift that really differentiates proteomics from microarray and sequencing analysis. We've already said that in the old days, sequencers could sequence up to 384 templates at a time if you had 384 capillaries in your machine. Today's sequencers, the massively parallel ones, are sequencing millions of templates at the same time. Microarrays, similarly, do not ask, is this gene being expressed? And then, is this gene being expressed? And is this gene being expressed? They're doing it all simultaneously. All of those probe sequences are competing to hybridize the cDNAs generated from cellular samples. Proteomics does not really operate in that paradigm yet. At present, almost all tandem mass spectrometry data is created by measuring generally speaking, a single tandem mass spectrum at a time, where each tandem mass spectrum represents one peptide blowing apart. In recent years, we have seen some methods that attempt to quantify all of the peptides present at a particular moment of chromatography. But generally speaking, those types of experiments are not intended to identify all the different peptides of a cell at one go. Generally, those are about quantifying instead. So, we're moving away from a scenario where all the data are collected in parallel to one in which the data are collected serially, one after the other. So if you have an instrument that can generate a very large number of tandem mass spectra in a given second, then you're going to have a better ability to get a very sensitive appraisal of all of whatever peptides are visible in the cell. Generally speaking, proteomics technologies are not as sensitive as sequencing technologies. If you want to measure gene expression for the best 90% of transcripts in a cell, you can pretty much do that with RNA-seq. But if you wanted to see 90% of all proteins in cells, you're going to need a lot of sample and a lot of instrument time. Very frequently, the case in proteomics is that we trade instrument time to accomplish more sensitivity. So if you're trying to do an experiment in a 2-hour block, you're going to see maybe a few thousand proteins, but you're not going to see typically tens of thousands of proteins. These numbers change from year to year because mass specs keep getting better. But I wanted people to have that appraisal on that. All right, so what is a mass spec? I really feel like this topic deserves a lecture all to itself. But I'm going to try to give you some of the basics to begin with. Do people remember the old tube TVs? Not like the flat panel ones, but like the ones with some depth and heft to them, right? You know, in the old days, if you were going to move your 30-inch TV, you brought in a buddy, right? Right? So one of the reasons those things are big is that they use magnetic fields to steer electrons, right? So you have a gun at the back of the tube that's firing particles that are then being directed to a particular location on the face of the screen to cause light to be emitted. And if they're directed at this part, you get a red light. If they're here, you get a green light. If they're here, you get a blue light. And so by rastering this beam of particles to each of these parts of the screen, we were able to produce a visual image to work from. A lot of how televisions work has a lot to do with how mass spectrometry works. We have a beam of ions that are being created. These ions represent the peptides, typically. We have a mass analyzer that's steering these ions by their mass to charge ratios. And we have a detector. Instead of bouncing them against a screen and emitting light, we're typically hitting them against a conversion dynode or something that can produce an electric signal, an electric current in response to how many ions are hitting it, or we're using something like Fourier transform. But these three pieces are shared, essentially, between your old tube TV and what a mass spec is doing. An ion source is creating ions of the peptides. The mass analyzer is allowing you to manipulate them. And a detector is allowing you to discern whether they've arrived or not. How much signal is available at a given mass to charge ratio. Mass to charge ratio shows up all the time. So if you hear me talk about M to Z, remember, we're talking about these characteristics of ions that allow us to manipulate them in the mass spec. Generally speaking, we're going to start our experiments with a protein mixture. Because after all, most biologists care an awful lot more about about proteins and what they're doing in cells than individual peptides, because in vivo, they function as proteins typically. So we start with a protein mixture, and one of the very first things we do on the bench is make that mixture much, much more complex. How many different proteins do we have in a given cell line? Kind of a puzzling question, isn't it? Are all of the proteins expressed to some level? Let's say we've got 100,000 transcripts in human cells, built from our 20,000 genes. Do we have all 100,000 of those protein sequences present at some level? Probably not. Probably not. Some of them are developmental, some of them only occur under very rare sorts of stimuli, blah, blah, blah. So, but would you say that the number of proteins in a cell is greater than or less than 1,000? Greater than. Anyone disagree? Are there some proteomic samples that would have less than 1,000 proteins? Definitely so. Right, so we often use things like co-immunoprecipitations and fire those through the mass specs. Typically, if those are relatively clean at least, they're not going to have 1,000 proteins in them. We may be shooting something very complex like a biofluid through a mass spec, and it will contain more than 1,000 proteins, but we won't see them all because of the dynamic range problems there. Lots of some proteins block the appearance of some smaller proteins. In something like a cell line, though, you could typically see many thousands of proteins, more than 10,000 proteins, in fact. So, in general, this peptide mixture may contain, let's say, 10 to the, let's say you've got 10 times as many peptides as you started in proteins. So you could have more than 100,000 peptides in these peptide mixtures. As I said, we frequently trade use of instrument time to get better sensitivity. So we might fractionate these 100,000 odd peptides into different fractions on the basis of isoelectric focusing, or on the basis of there are lots of different methods that we use for this. Strong cation exchange affinity, stuff like that. Lots of different ways. So these 100,000 peptides have now been separated into mixtures that maybe contain 10,000 peptides apiece. We use liquid chromatography, typically reversed phase liquid chromatography, to separate out the peptides out of one of these fractions. Right? So we do the first vial, then we do the second vial, then we do the third vial, each of these taking maybe 90 minutes of experimental time. So this liquid chromatographic gradient in a 90-minute experiment is usually spanning about an hour, where we gradually raise the level of hydrophobic solvents to knock peptides off of C18Bs. So the peptides fall off of the column, and then they come out of the tip of the column, but once they reach the tip, they're not going to drip off. Instead, we're applying maybe 3,000 volts to the end of that column, which causes these peptide ions to become gas phase ions. And they flow into the mass spectrometer where they can be analyzed. So all of these steps at the top are taking place outside the mass spec. The bottom row, however, is all taking place inside the mass spec. And we're going to measure these peptides that are appearing in solution in two different ways. And remember that the liquid chromatography is going on for about an hour, and the mass spec is doing this work repeatedly for each of these different instances of time throughout that gradient. So first off, we're typically going to use high-resolution mass spectrometry, maybe Fourier transform mass spec, in order to get a very, very accurate measurement of the precursor ions. Precursor ions. We call them precursor ions because we're interested in tandem mass spectrometry, but what you need to remember is that a precursor ion here is a peptide. Maybe this peptide has one proton sitting on it, in which case it's a +1, or it may have 2 protons on board. It's a doubly charged precursor ion. Maybe it's carrying 3 protons if it's really big and has lots of basic residues, right? So these ions that are being created in the process of liquid chromatography are going to have very, very accurate measures of their mass-to-charge ratios made through this first round of mass spectrometry. That generates a mass spectrum. We're going to talk about the difference between mass spectra and tandem mass spectra a little bit more. But I mentioned that we're measuring them 2 ways. The first way is to look at them intact, to say what is the mass-to-charge ratio of the peptide ion. The second way is that we're going to look inside them. We're going to interrogate the structures of these peptide sequences. For that, we're going to isolate a whole bunch of ions that all fall at the same mass-to-charge value. So that the software is able to look at these mass spectra of peptides, intact peptide ions, and say what ions dominated, had the best intensity signals at this particular moment in time. And then it says, okay, these 5 all look good to me. I'm going to isolate this one first, and then that one, and then that one, and then that one. And each of these will be subjected to tandem mass spectrometry. So having isolated a bunch of ions of a particular peptide, it uses gas, collisions with gas specifically, to energize those peptides. This energization process causes peptide bonds to break. This is the process of collision-induced dissociation, or CID. So having created a bunch of fragments for many, many copies of a particular peptide, we then analyze the mass-to-charge ratios of the fragments produced from that peptide. So the mass spectra tell us which peptide ions were available to break at this point in time. The tandem mass spectrum shows us the list of fragments that were created when we blew apart a particular peptide. In the course of a single 90-minute experiment, I will probably generate tens of thousands of tandem mass spectra. Generating even 10 tandem mass spectra a second is possible with current mass spectrometers. So we can generate huge volumes of data, but it creates a big problem trying to figure out what it all means. Now, I've mentioned a fair number of disruption steps along the way, and I want to try to walk through this a little more slowly so that people get those pieces. First off, proteins have kind of a complex structure, you know? They're all bundled up and they have these disulfide bonds holding them in place, and they have a lot of structure, and most proteins are kind of resistant to proteolysis, chopping them up. So one of the things that we want to do is to denature these proteins, to cause them to stretch out and to reduce disulfide bonds. You know, if you're trying to unfold a blanket and it's all held into a jumble by a bunch of safety pins, you probably want to take those safety pins out before you try to flatten the thing. Proteins are the same way. We want to cut these disulfide bonds as we denature the proteins to make them linearize nicely. At that point, we're going to chop them into peptides, typically through enzymatic cleavage. Now, in proteomics, almost everyone ends up using trypsin at some point. So trypsin is a special enzyme that cuts only after arginine and lysine residues. At least we ideally say so. So the denaturation and reduction step is one that takes place for intact proteins. Enzymatic cleavage starts with intact proteins, but produces peptides. But it's only after those peptides are in the mass spectrometry environment that collision-induced dissociation can take place. So these first 2 processes take place outside the mass spec. The third one takes place inside. Any questions so far? Okay. So I've mentioned that we have mass spectra and we have tandem mass spectra. So the peaks that we see in a mass spectrum represent the peptide ions that are currently eluting from LC-ESI. We love our jargon, don't we? So LC is liquid chromatography, the reverse phase liquid chromatography. ESI is electrospray ionization, this process of zorching the ions coming out of the column. I always think of it as a bit of a cattle prod attached to a water hose. But anyway. So when we look at this, we are looking at a mass-to-charge axis. So the ions are being illustrated across that mass-to-charge axis. And then we have an intensity signal. Now this is not abundance. It's not abundance. So if we see that the ion at 735 produces a big peak, and the ion at 582 produces a small peak, there's some temptation that you would say, there must be more of this ion at 735 present than at 582. That is a wrong inference, because each ion has a different kind of potential for ionization. Right? If you have 1,000 copies of whatever this is and 1,000 copies of whatever that is, you've no guarantee that they're going to produce similar spikes on our mass spectrum. So generally speaking, we can only compare this signal for 582.97 with another signal for that same peptide. Okay, so this mass spectrum then says to us we have an ion available for interrogation at 735, one at 645, 609, 767, etc. These are available now for us to interrogate. It's only after a particular ion has been selected from this, isolated, beaten up by pouncing it against a bunch of gas ions, and produces fragments, that we get a tandem mass spectrum. The tandem mass spectrum then is a list of fragments, but it is also, at least in the kind of shotgun proteomics techniques we're talking about, it has the information of what we blew up to create this set of peptide ions. So we remember how big what mass to charge ratio was characteristic of the peptide that we blew up, and we have the list of fragments that we're seeing for it. This tandem mass spectrum is our source material when we're doing our identification of peptides and proteins. There is a common misperception among people in proteomics that the art of CID is basically to blow up the peptides. You know, just zorch a whole bunch of energy into this peptide, and then suddenly the peptide bonds are all snapping willy-nilly. This is not actually how things happen. Generally speaking, there's a brilliant body of work on this subject from Vicky Wysocki and others on the mobile proton model that explains this process in a lot more detail. Also a really great paper from a guy named Bellville, a very nice gentleman, that talks about the pathways and competition model that explains this process. But in general, what we have is something that looks a little like this. So what do we know about protons? Where do they like hanging out? Protons are positively charged, right? So they like sites of high gas phase basicity. So something like arginine is home sweet home for a proton. It's like a big catcher's mitt for protons, frankly. Lysine is not as basic as arginine, but it's still pretty basic. And then, of course, we have the peptide N-terminus. All of these are pretty comfortable places for a proton to sit. But when you start adding energy to a peptide, you start an exploration of the space. And suddenly, that proton may find itself associating with the oxygen on a carbonyl. So weird things happen when you do that. When you park a proton up here, these electrons kind of cheat in the direction of that proton, making this carbon a little bit positive. And when that happens, the pair of electrons on this carbonyl can attack across there to form a ring, and whammo, suddenly you've got the breakage of this weird intermediate into these B ions representing the N-terminus and Y ions that represent the C-terminus. So we've been talking about 5', 3', and I was in everybody's comfort zone. Now we're talking proteins. So the left side of the protein sequence is the N-terminus, the right side is the C-terminus. The left side produces the B ions, the fragments that contain the N-terminus are the B ions. Those that contain the C-terminal side are the Y ions. So Y ions have exactly the same structure as little peptides. See that? NCC, NCC, that's 2 amino acids. You have a little side chain coming off here, a little side chain coming off here. This is the same structure as an ordinary peptide. But B ions are a little weird, aren't they? NCC, but now we've got to go NCC around that ring to recreate the structure. We started with just a standard 4 amino acid peptide. So this process is not a blasting apart. It's the energization of peptides, and the proton gets adventurous, moving places that are a little more uncomfortable for the peptide, and so suddenly one of these peptide bonds breaks as a response to that. So how can all these peaks tell us a peptide sequence? That is, I think, a difficult point for a lot of people to follow, so I want to try to walk through this again. So let us imagine that we have a 10 amino acid sequence. Does everyone remember their single letter codes? No! Oh my goodness. Well, if you're my grad student, I'm going to quiz you from time to time about what's the 3-letter code for proline? It's pro. Okay, that's not so bad. Anyway, so we've got threonine, serine, isoleucine, glycine, threonine, isoleucine. I can go on. But each of these has a characteristic mass to it. All right. So if I were to single out a particular student in the room and say to them, what is the lightest of all amino acids? She would look frantically in another direction. Oh, okay. It's good for me to ask these questions because I don't want to just assume people know stuff. All right, the simplest amino acid has what as its side chain? What's the lightest element? You can say it. Lightest element is? It's number 1 on the periodic table. Did I hear hydrogen? That's very good. Okay. So the very smallest side chain among all of the amino acids is that of glycine. It's a hydrogen. So NCC, the C in the alpha carbon here just has a pair of hydrogens flanking it, and the C is just an ordinary carboxylic acid. Okay, so glycine is the smallest of amino acids. We've got a glycine right here. Glycine contributes 57 mass units to the mass of the peptide it's in. What about the biggest one? Does it have a hydrogen as a side chain? It's got a lot more than that. All right, we've got tryptophan, right? 184 mass units. So 184 versus 57, that's a pretty big mass difference, right? We can easily tell that apart. Now, some of the most popular amino acids that we find in structures are isoleucine and leucine. And they're a complex case for proteomics because they have the same mass at 113. All right, so now we have a few numbers to kind of guide us in understanding this. If I have a breakage event in this peptide sequence, then the 1, 2, 3, 4, the fourth peptide bond in the sequence is sheared, is broken in the process of collision-induced dissociation. That means that we have 4 amino acids, 1, 2, 3, 4, that are part of, that contain the N-terminus. That is a B4 ion. So what does that make the other 6 amino acids? It makes them the after ion. Of course it does. Before and after. Oh, I love that joke. Anyway, so these 6 amino acids together depart as a Y6 ion. B4 because it's got the 4 amino acids and it contains the N-terminus. This is the Y6 because it has 6 amino acids and contains the C-terminus. We see evidence for where those appear in the tandem mass spectrum. If I add together the mass of T, S, I, and add a little offset, I get to 415.2. So this ion is consistent with the appearance of a TSII B4 ion in the spectrum. The Y6 ion is the mass of GTI GPK plus a little offset plus water, and it comes in at 572.3. Great. So evidence for this particular peptide bond break is coming from both avenues, from the N-terminus here and from the C-terminus here. All grand. Now, is it easy for you to simply read this spectrum and infer what sequence created it? I spent a whole semester-long course learning how to do that, and let us describe that process as difficult. Right? De novo sequence inference is feasible. You can even do it with software, but it is not straightforward, and it's not for the faint of heart. It requires a good calculator and patience. So, we would like not to try to infer these sequences, but I'm going to go one step further in trying to show you that these peaks that we see here are indicative of the sequence. Imagine that our breakage was not after the 4th amino acid, but instead after the 3rd. So TSI then break. What would happen to the mass of the Y ion, that's the Y7, versus the Y6? What's the difference in mass? If the breakage happens here rather than there, the Y ion gains isoleucine. Everyone sees that? Isoleucine has a mass of, somebody wrote it down, 113. So 572 plus 113 is 572 plus 113. 685. Look at that! You did it. You found the next ion. Right? So we can look at the fragment ion evidence to confirm or negate the claim that this spectrum represents that peptide sequence. Okay. So we can confirm whether these tandem mass spectra in fact represent peptides, but it's rather hard to start from a tandem mass spectrum and guess what peptide sequence it is. This brings on the database search algorithm. Now, the database search algorithm is old like me because it was first published in 1994 when I was a sophomore in college, a second year in college. Right. So, I'm going to try to contextualize how this whole pipeline fits together. The process of starting with a collection of tandem mass spectra and coming up with a set of raw peptide identifications, creating peptide-spectrum matches, is the process for the database search engine. We are also going to talk about how we discern which peptides we actually want to keep from a collection, and how we infer a set of proteins from that. But most of this lecture is about this step right here. Given a bunch of tandem mass spectra, how do you discern what peptides you would claim those spectra represent? All right, this block diagram is probably the most useful thing to take away from this lecture, because this shows all of the pieces that we include in our database search tools. We start with a sequence database. Now, we already talked about FASTA sequences in one of the earlier lectures this week. A FASTA sequence database is a list of protein sequences, or a list of nucleotide sequences, that each have some accession associated with them and some description associated with them. Generally speaking, if you're doing clinical proteomics, you're going to use a FASTA sequence database that contains every protein sequence that we know can be found in humans. All right, and generally that's just going to be the reference sequence. So if you have a valine rather than a leucine at some position in a given peptide, you would fail to identify that if your sequence didn't match the canonical sequence database. Generally, we ignore that. So we start with maybe 100,000 protein sequences for human. We probably add some contaminants. What is a protein that is likely to show up in our samples even though it's not in the sample that we started with? We already talked about it for digesting up the proteins. Trypsin, right. So we need to include some contaminant sequences that are likely to have been introduced by the sample handling, including porcine or bovine trypsin, depending on which one you use, is a first step. If you are working with yeast guts, are you only going to find yeast proteins and trypsin? No, because very frequently we've done stuff like run our samples through gels, and if you run stuff through gels, you're going to find loads of keratin in there. Those buffers, I swear, they're just the dirtiest, awful things. If somebody had dandruff near you on the bench, boom, bingo, suddenly you've got a bunch of human keratin, right? So, yeah, I don't mean to single out people with dandruff. I am known myself, but in any case, we end up with some proteins there that we didn't think were in our samples. We add those as contaminants along with all the different proteins that are possible for the sample, for the origin of our sample. That provided a bunch of protein sequences. But remember, what we're identifying directly are peptides. So in silico, we need to do some of the same steps you just did on the bench. You digested up your proteins on the bench, therefore in silico, we need to have a function that acts as a trypsin digester to produce lists of peptide sequences from those. Now, part of the Sequest, the first database search algorithm that was not published in the first paper, but was picked up in the second in 1995, is this PTM expander. Does anyone know the phrase PTM, where that comes from? Post-translational modification. Lots of proteins bear modified masses. Now, I would note that one of the most popular ones that people look for is phosphorylation. Phosphorylation can appear on serines and threonines and tyrosines, and it adds a mass of 80 to create a different mass for phosphoserine than for serine, or a different mass for phosphothreonine versus threonine. The mass difference is 80 in both those cases. So the PTM expander starts with a bunch of peptide sequences and then decorates them with all these sites where modifications may have happened. Generally speaking, those rules are very simple. They say something like, if I see a methionine, imagine the methionine is at its normal mass or at 16 higher, because methionine gets oxidized if you just look at it wrong. If you are looking for acetylation on N-termini of proteins, then you would specify that the N-terminal peptide can have a mass of 42, higher than what we would normally expect. Those are examples of PTM expansions. So these decorated peptides are now ready for us to go about comparing to tandem mass spectra. A very, very important feature, really the reason why Sequest was able to work in the first place, is that we don't need to compare every decorated peptide to every tandem mass spectrum. What information did we collect about each tandem mass spectrum beyond the list of fragments present in it? Do you remember the dashed green line in the tandem mass spectrum a couple slides back? That is a precursor mass to charge ratio. So generally speaking, we know the mass to charge so accurately that we're able to tell its isotopes apart and even infer the charge for the peptide. So knowing a peptide mass means that you don't have to compare every peptide to every spectrum. You only need to compare those peptides to the spectrum if they're very, very close to the mass of the whole peptide. That's magic. So this mass filter gives us a list of candidate peptides. These peptide sequences, we can predict what the spectrum should look like if it were this peptide, and then compare that set of predicted fragments to what we observed in a match scorer. Okay, all of this was published back in 1994, and then modified in 1995 to include PTM hunting by my PhD advisor, which is one of the reasons why this has been something I've been talking about for 20 years. All right, on we go. So, how do you go about emulating the behavior of trypsin in silico? Imagine that you have some protein sequence. Trypsin isn't going to cut just anywhere, typically. It's going to cut at arginine and lysine residues, which means if you look at the sequence of that protein, you can predict sites that are most likely to be locked by a trypsin. Some of these peptides that would result stretch from a legit trypsin site to the next legit trypsin site. And that gives us what's called a fully tryptic peptide. Those are the easy ones, right? Because they are trypsin behaving just like you want it to. Sometimes we find that other things are going on in our samples. We may have other proteases. We may have the voltage up a little high, and so some of the peptides are getting blasted apart as they go into the mass spec. This can result in semi-tryptic peptides that conform to normal trypsin behavior on one end, but not the other. And sometimes, trypsin doesn't chop everywhere you expect it to. Sometimes you have an incomplete digest. Maybe it's because this protein wasn't unfolded completely. Maybe you cut short the digest a little bit. Who knows? Lots of reasons why. So that can create a missed cleavage peptide. The other possibility is that you might have a stretch of this protein sequence that runs through a membrane or something, a big transmembrane domain. Those dudes don't have a lot of arginine and lysine in them. As a result, you have this long stretch of undigested peptide that you need to be able to model. Anybody who's ever done a database search with post-translational modifications is aware that it's a lot slower to use lots of modifications than not to do so. The rationale for why is sometimes a little hazy though. So I've grabbed a peptide sequence from alpha-casein in cows, DIGSESTEK, as an example of how a phosphorylation search can blow up the search space, can give us a lot more decorated peptides to consider. So in this top case, we have the peptide as drawn from the sequence database. In the second case, we have a star, an asterisk here on the serine. That is to indicate that this serine has been modified, in this case by 80 mass units, because it's got a phosphorylation on it. But this serine is not the only place where this peptide can be decorated. This second peptide, or the third one in the roster total, has a star on the second serine, not the first. And then in the third outcome, we have a threonine that's been modified this way. These three peptides, all the red ones on screen, have the same mass, because in each case, it's the original peptide mass plus one phosphorylation. They do, however, have different tandem mass spectra, because when this modification moves from residue to residue, it alters the masses of a few B and Y ions that would contain those masses. The next set of blue peptides are the same sequence again, but this time with two different modifications on board. Both of the serines here are phosphorylated. In this case, the first serine and the threonine are phosphorylated, and in this case, the neighboring S and T are both modified. Again, these blue sequences have identical masses, and that mass is 80 units higher than these red ones. These red ones, in turn, are 80 mass units higher than the black one. And finally, the green sequence at the bottom has 3 modifications on board, so its mass is a total of 240 Daltons higher than the original mass of that peptide. In this case, we have an exponential expansion of our search space just by allowing for serine and threonine to be phosphorylated. And typically, we also allow for tyrosine to be phosphorylated, so it's even worse than this. So in this case, you can think of this as a branching path. As we work our way through this sequence, when we hit this first S, we have 2 possible paths we could follow. Is phosphorylated, is not phosphorylated. When we hit the second S, we have another branch in the path. May be phosphorylated, may not be. When we hit this third, the threonine, it may be phosphorylated, it may not be. Which means that with one peptide, we now have 8 paths coming out of it. Which is the same thing as 2 possible outcomes for these residues raised to the 3rd power, because there are 3 of them in the peptide. I brought in some math, I'm sorry, but it's helpful to be able to model this. Essentially, the longer the peptide you've got, the more modifiable residues it's got, the greater the likelihood that you're creating this blast of different possibilities as you allow for modifications. People use a lot of different mass specs to do tandem mass spectrometry. I talked about high-resolution mass spectrometry measurement of peptide ion masses, and I showed you a little picture of something that we call an Orbitrap, which is one of the most widely used instruments for this. There are, however, lots of people doing mass spectrometry and tandem mass spectrometry on simple ion trap instruments. They're certainly a lot cheaper, and they don't die. So quadrupole ion traps, on the other hand, don't tend to have very good mass accuracy. Having measured a peptide, you don't know its mass within 0.001 Daltons. You know, that's feasible with an Orbitrap, but it is not feasible with an ion trap. So typically, we have to have a very wide mass window, a tolerance that we use in deciding which peptides can be compared to each spectrum. If you use something like an LTQ or a Velos instrument, you're going to have a lot more time required for identifying peptides, because you have to compare many more peptides to each tandem mass spectrum. So this is not a favored approach among labs that are doing proteomics every day. You see a lot of people today using quadrupole time-of-flight instruments. For example, we have a triple TOF over at UCT that people use. There are a fair number of these in the country at this point. QTOF instruments have much more mass accuracy than quadrupole ion traps. And I've written that you can assume a mass error of 100 ppm parts per million for their precursor mass accuracy. That's easily within 0.1, and more typically within 0.01 of the true mass of that peptide. 100 ppm, frankly, is an underestimate of how accurate today's mass analyzers are from the QTOF family. And finally, it's quite typical that people are using instruments like the Fusion or the Orbivelos over at our campus, and those allow for a 10 part per million mass accuracy from these peptide ions. If you can draw it this narrowly, the mass accuracy options are really quite stellar. You know the mass at that point within 0.002 Daltons. It means that very few peptide sequences need to be compared to each spectrum. Great, so we've talked about how to digest a protein in silico. We've talked about the impact of post-translational modifications on how many candidate sequences must be compared to each spectrum. And we talked about the peptide mass filter, which is the feature that allows us to decide which peptide sequences get compared to each spectrum. But we haven't really talked about how to compare an observed tandem mass spectrum to our model of it. First off, it's probably worth saying something about how we predict what a tandem mass spectrum should look like for a given peptide sequence. Right? So we have a peptide sequence. We can imagine then, if we chopped after the first amino acid, what would B1 look like? Now, that doesn't really happen in nature, so let's move on to the next one. Let's say we chop the second peptide bond of this peptide. We can compute the mass of the B ion that should result from that, because we know what those first two amino acids are. Therefore, we can say the B2 ion should appear at this mass to charge value. Then you move to the next peptide bond. What if it broke after 3 amino acids? What would the mass of the B ion be that came out of that? What would the mass of the rest of the peptide sequence be that came out of that? So we can predict all these mass to charge values of the fragment ions that should result if it were this peptide sequence. And that might be a very simple model. We might simply say, on the mass to charge number line, I expect a peak of some height here, 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 and here. That's a pretty simple model. You might have software that tries to say, these ions, being Y ions, should be taller than these ions, which are B ions. Some might even try to take sequence-specific effects into account. For example, proline, when it breaks much more readily on its N-terminal side than its C-terminal side. But frankly, most software doesn't go that far. It mostly says, these ions should be Ys, these ions should be Bs, and it may model different heights for the two. Okay, so let us now imagine that I have a spectrum that I have observed, represented by my right hand. I have a second spectrum that is theoretical. I have some peptide sequence, and I've said, I think this is where ions should appear if it were this peptide sequence. The process of cross-correlation obviously has some math involved, and it's morning, so this is your best shot probably at absorbing the math today, but I'm going to give you a simpler way to think about it. The theoretical spectrum and the observed spectrum are rubbed past each other. Shoot, just like that. And at each of these offsets, each of these different relative sliding positions, we're going to ask how well does the signal that we observed correlate with the one that we predicted for this particular sequence. Do they offset well here, 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 here? This hairball that you see at the lower right represents the score at each of these offset positions. Tau 0 means that the 1,000 that we observed is aligned with 1,000 that we predicted, right? So tau 0 means there's no offset between the two. And you see that there's a big spike in the middle of that hairball. The reason why Sequest does this is that given an arbitrary positioning of these two spectra relative to each other, there's some amount of random matching that takes place. And the software wants to say, how much better is the match when there's no offset than when there's some offset? So having generated this hairball, essentially Sequest is saying, how much bigger is the spike in the middle than the hairball around it? That is a Sequest score. Now, I note that I've got 2 publications listed here, 1994 and 2008. I think everyone remembers that 1994 is the year that Sequest was originally published. We just talked about that. But 2008 was this interesting paper from the same guy who had written the original source code for Sequest, Jimmy Ng. Nice guy. we could tell stories. Anyway, Jimmy is a very funny guy. So in 2008, he published this paper in which he noted that there was a math simplification error in the original 1994 publication. And after simplifying the math for Fourier transform, which we talked about before with Fred, shows up here again, after performing this correction of his math, the software was able to run 10 times as quickly. Now for all the people who wasted their time back in Pentium 3 days running Sequest, this is irritating. Imagine if we could have had our search results back 10 times as quickly. Very vexing. Anyway, he did the math correction in 2008, and another search engine has resulted from that. So the Comet search engine is also freely available for everyone now. So the cross-correlation approach has shown up in a whole lot of different search algorithms out there. I wrote a paper about the family tree of Sequest that relates to all of the inheritors of this approach. Now, if you spend any amount of time over in the Blackburn lab over at UCT, you are probably going to encounter someone talking about their MaxQuant search results. MaxQuant is not my favorite search engine, I have to say. I have a bit of a bone to pick with some of the approaches they've chosen, but my feelings don't really come into this. This is a good example of a match counter scoring approach. Before we talked about rubbing one spectrum past the other and evaluating the alignment. We're not going to do any of that. Imagine instead that you have a jar of marbles, and this jar of marbles is mostly white marbles that reflects a place where there could have been a peak in the spectrum, but there wasn't. And it's got some black marbles in there too. Those black marbles represent places where there could be a peak, and there was one. All right, so let's say that you've got 9 times as many white marbles as you do black marbles. Right, so the spectrum is mostly empty, but it's got a few hundred of these sticks in it. So if you imagine the predicted spectrum as an M over Z line that just says these locations should be occupied and these others shouldn't, you can think of that as being a draw on your jar of marbles. That everywhere you predicted there was a spectrum, you draw out the marble corresponding to that M over Z location. And then you look in your hands and you say, well, I went looking at 20 locations in this spectrum for a peak, and 15 of them I observed a peak. What do you do with that? Is that a good score or is that bad? One of the things that we need to do is have statistical models that allow us to evaluate how unlikely such a thing is to take place by random chance. In this case, they use the binomial distribution. I'm sure you all adored your statistics class so much that you went home immediately and flipped a coin about 20 times to see whether it was a fair coin or not, right? The binomial distribution is our best friend for stuff like that. So you can see that we have math here. I'm not really going to beat you to a pulp on it. I'm just going to say that the software wants to ask the probability that we matched, in this case, 15 of the locations to a matched peak, or more by random chance alone. So we're computing a p-value, essentially, to say, is it at all likely that we would get this or a more extreme result than what we saw? Now, it has some oddities to it. One is that some peptides throw off water or throw off ammonia from their fragment ions. So the software asks, if I build those in, if I say this peptide has an oxidation on methionine, does it also lose a particular mass associated with that? Then, if that makes the score go higher, it's going to accept that those modifications, or that those losses should be modeled in the spectrum. Something else that it does that is very similar to what's used in the mascot search algorithm is that it asks, how big does something really need to be before we call it a peak? Right, so in a given tandem mass spectrum, maybe we have 200 locations that have some amount of intensity, but most of that is way down in the grass. So a lot of search engines say, you must be this tall to enter, right? Unless your intensity is above this, you're not a peak, you're noise. So the software is attempting to find the best place to tune that intensity cutoff in order to maximize its score. So this is the Andromeda search engine built into MaxQuant. That's how it does its thing. It's very different than Sequest, isn't it? Right? I mean, none of this cross-correlation business going on at all. This is just a match counter by comparison. So there are a bazillion other search engines. I'm not going to walk through them. In fact, we're going to take a break right here before we go on trying to figure out whether we can trust the result from our search engine. And welcome back. All right, database search was very exciting indeed. But it is time for us to revisit the topics of false discovery rate, because we need to learn how to know which PSMs, which peptide spectrum matches are trustworthy and which are not. So when I'm going to use PSM a fair bit, I want you to remember that they represent a hypothesis about a tandem mass spectrum. We hypothesize that this tandem mass spectrum represents this peptide sequence. That's a peptide-spectrum match. All right, so how do we decide which ones are trustworthy? Now, I've already mentioned database search scoring. And the goal in that first step, the goal of the database search algorithm, is to make its best hypothesis about what each tandem mass spectrum represents from the peptide database. So it may have a high score associated with it, or it may have a low score. But what the search engine tells us is that this peptide, drawn out of the whole sequence database, was our best guess at how to represent this tandem mass spectrum. Now that could be right, or it could be wrong. Imagine, for example, that this tandem mass spectrum represents a peptide from chicken, because we're looking at my gut proteome. And yet you didn't look for chicken proteins when you searched my ruminant sample. So it could be that that peptide has a beautiful spectrum, but because the chicken proteins are not in the database, you don't recognize it. In a case like that, the search engine is going to do the best job it can for explaining why that spectrum looks the way it does. It's going to put some peptide sequence up against it. But its score should indicate that we don't have the right sequence associated with it. So that's a really important lesson for proteomics. If it's not in the database, your search engine isn't going to identify it. I kind of wanted to go old school and use the word, it ain't gotta be there. There you go, it ain't. No one says that word here, right? Okay, well, there you go. I lived in the South for a long time. So, the database search engine scores then need to cut in two different directions. We want the database search engine to use those scores to find correct sequences rather than incorrect sequences. So it's got to figure out what one correct sequence matches so much better than everything else in the database. That's the first role for it. In the second case, we're going to use it across spectra. We're going to say, of all of the spectra that we identify to some sequence or another, which ones do we trust and which ones don't we trust? That's this application on the right. We want to filter our peptide spectrum matches to include just those that are real and exclude those that are not. All right. So how do we control this error? There are two big approaches that get used most frequently. I think many people now are accustomed to using systems like the Trans-Proteomic Pipeline, the TPP, or scaffold software. These are really popular worldwide. They use an approach called distribution modeling to decide what the probability of error is for individual PSMs. So rather than say, out of this set, I think the accuracy is blah, 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 they put a probability of error on every PSM. Okay, that's a very specific kind of goal. In the second case, in Target-Decoy search, we're most typically trying to make a statement about the collective identifications. Out of this set of IDs, we think the fraction that's false is about 5% or about 2%. All right, so these represent kind of different goals, and they also represent different approaches. So let's start with PeptideProphet. PeptideProphet was published by Keller and Nishvishki back in 2002. I think they were both postdocs back at the time. Alexei Nishvishki is now a very senior professor in the space of bioinformatics for mass spectrometry, a good friend too. So let us imagine then that we're looking at IDs from Sequest. Does that sound like I'm throwing his name around? I don't mean to be vainglorious like that. I'm very lucky he's my friend. I'm aware of this. All right, so Sequest is a database search algorithm, and it outputs several different measures to say how much it liked this peptide sequence versus that spectrum. We have the X-core score that represents the rubbing of one spectrum past the other, how much that spike stands out from the hairball. It has a Delt-CN score, which represents how much better the best match is than the second best match. That's important too. Do you want that number to be high or low? Do you want the best score to stand out a lot from the pack or a little bit? A lot. right? So if I'm running a 100-meter fastest man in the world challenge, I want to leave number 2 in the dust 5 seconds behind me. That would be impressive. If I'm 500ths of a second ahead of the next person, well, I still win. But that would be impressive to see me ever win a foot race. In any case, that's the interpretation on this delta-CN score. It also has something called a preliminary score rank. Sequest has multiple scoring engines that it uses to compare, to score these matches. So it preserves information about that in the rank, where this peptide showed up in the original preliminary score ranking. DM probably looks like a kind of uninformative score, but in fact, it's very, very useful indeed. You see, if you're measuring the peptide ions mass to charge value, it may be very close to what the peptide sequence says it should be, or it may be relatively far away. This is delta mass. So the search engine score, the cross-correlation score or the binomial score that Andromeda uses, that doesn't really take into account whether the peptide ion was measured to be close to this sequence's mass or far away. So Nesvitsky realized that he could compute the delta mass and use that as another score. If they're very close, give it a bonus. If they're very far apart, give it a detriment. All of these things could be combined together to give more information than any one score by itself could achieve. And he called that a discriminant score. So the discriminant function tells us that we will use these weights on these scores and use that number to rank our IDs. If we then look at all the PSMs that came out of a particular search, we get 2 different distributions put on each other. Now, first off, I want to point out that this paper came out in 2002. A lot of mass spectrometry has gotten a lot better since then. But back in those days, it was quite typical that out of the 1,000 or 10,000 tandem mass spectra you had collected, you would identify only about 10% of them successfully. That's shocking, isn't it? Imagine if you did a sequencing experiment and you could only map 10% of the reads to the annotation. That would be kind of embarrassing. You would want to know what's going on with that. But in the bad old days of early mass spectrometry, we frequently had problems getting enough ions into the trap to get a good tandem mass spectrum from it. And we had poor enough mass accuracy that we were comparing a lot more sequences to each spectrum than we really wanted to. So what we're seeing is that we have 2 distributions. This hump over here represents the set of PSMs that have accurately been identified. This big hump down here represents the set of PSMs that have been incorrectly identified. What we have is a mixture model situation, where we need to be able to say, given a discriminant score, do things belong in this tail up here on the high end, or do they belong in that big hump down at the left? So given a discriminant score, you can ask, say for a score of 3, what is the height of the positive curve here? What is the height of the negative curve there? And using that information, you can compute a posterior error probability to say what is the probability of error for this particular PSM. Kind of magical. And it's not rapid either. If you've used TPP for your PSM evaluation, you know it's going to take a few minutes to run through all of that, or much longer if you have a very large set of spectra that you're trying to make sense from. So this process gives us the ability to evaluate the error probability for each PSM individually. Very powerful, especially if your information that you're going after is correctness of this particular phosphopeptide, for example. All right, now let's look at an easier approach. This is one that gets used all over the place, so it's worth knowing about. Let us imagine that we have some score to represent each PSM. Maybe it's just the search engine score. So you can imagine that I've got them stacked under my hand here. Way here at the top, I have the best scoring PSM out of the entire raw file, the entire gradient. Down here on the floor, I have the very worst PSM we saw, the very lowest scoring one. Now, if I care about having no false positives at all, I'm going to lower my bar by 1 PSM and say only the best scoring PSM out of this raw file is correctly identified. Now that's nonsense, right? I mean, we wouldn't do that. Why? Now, my false discovery rate at that point is essentially 0, and I've got a great ID, so why do I want to go further? Because sensitivity matters. If I push this bar lower, yes, I may add some erroneous PSMs, but I will certainly pick up some that are real. So I need to find how far down I'm going to push this bar, how much error I'm willing to tolerate, so that the PSMs over that bar give me good sensitivity, but not an excessive false discovery rate. Okay? Now, how can we accomplish that? When we run our database search in Target-Decoy mode, we don't just include real protein sequences. We also include decoys, sequences that we know are not represented in this sample. One of the most common ways that people do that is called a reversed search. So I might have, for example, the human protein sequence for actin. It's N-terminus here, it's C-terminus here. I now include actin 2 ways in my database search, in its normal orientation, and then I reverse the series of letters from C-terminus to N-terminus and stick it in there. Furthermore, I'm going to mark that as a decoy protein. So maybe I started with 100,000 protein sequences from human in my database, and now I have 200,000 sequences in my database, but half of them have been marked as bogus proteins. This is not real, it doesn't exist in nature. So, let's return now to our stack of PSMs, and I'm trying to figure out where to set this bar. If I lower the bar, and I accept into my good set a spectrum marked decoy, have I added a false positive or a true positive? It's marked as a decoy, which means that this peptide sequence doesn't exist in nature. But the search engine still said this was its best guess at what that spectrum represents. So if we lower the bar and we include a decoy sequence in there, we know we've accepted a false positive into our findings. Now, what if I lower the bar and I add to our collection a PSM that is associated with a target sequence? Do we know its status? We don't actually know. Because that sequence does exist in nature, it's possible that it was correctly identified. But it's possible that it was wrongly identified to a sequence that does exist in nature. That leads to a little bit of oddity in the math. So generally speaking, when we lower the bar and have some set of IDs, we know how many of them came from decoys, we know how many of them came from targets. We're going to assume that for every decoy sequence in that set, there's another false hit masquerading among the true or target sequences. Okay, so for every known bogus sequence, we assume there's an unknown bogus sequence hidden among the targets. So what this is going to give us is not which PSMs are right and which PSMs are wrong. It's giving us the ability to estimate the error rate for this collection of IDs that we're calling acceptable. Now, something that we frequently don't talk about in proteomics is that some of the spectra outside this range were also correctly identified. But because accepting them requires us to accept too many decoys, we deem them as lost. So we may have a much larger number of spectra that were correctly identified that fall outside this set, but the cost of accepting them is to produce too high an overall discovery rate. Those are false negatives. Those spectra have been deemed to be falsely identified even though they are real. So that's an uncomfortable place. Today, it's quite typical that if you're using a data set with high mass accuracy precursors and high mass accuracy fragments, collected from samples in which you have enough protein quantity, that you can identify 50% of the spectra that you collect. But it's still the case that there's going to be half of your data that can't be confidently matched to a protein sequence. It's an awkward place, but it's where we are in the field. It's a whole lot better than it was a few years ago. Now I'm going to return to a theme that I started with earlier. So we are not directly identifying proteins. We are directly identifying peptides, which means that if somebody gives you back a protein list from your proteomics experiment, they are giving you an inference about which proteins are the best explanation for the peptides we observed. That is a little bit uncomfortable for people. So if you really care about which isoform from a particular gene is represented by your data, you'd better be prepared to interrogate it pretty closely to get that information out. Shotgun proteomics produces tandem mass spectra, that's an MS-MS, that represents individual peptides. We are inferring a set of protein sequences that best explain the peptides we've seen. The MS-MS is produced from a peptide that has no memory of which protein it came from. So if you have multiple proteins in the database that are all equally good explanations for where this peptide came from, you'd better consider them all. If you are interested in understanding which, for example, phosphorylations appear together on a particular protein sequence, you'd probably better use something like top-down proteomics. I'm not aware of a lab in South Africa at this time that's doing top-down, but there's a consortium for top-down proteomics based out of Evanston at Northwestern University in the United States that does some really amazing work in this space. Lloyd Smith and friends published a really good paper introducing the term proteoform to help explain how this technology works. All right, so why is it that the sequence database may contain multiple proteins that could possibly be the origin for a given peptide? It may be that you're using a multi-species database. Has anyone here ever looked at SwissProt or UniProt for that matter? Right, so UniProt, SwissProt is one of the most commonly used multi-species database. If you have a peptide from actin, for example, and you've identified it versus SwissProt, SwissProt will have a lot of possible proteins from a lot of different species that represent, that contain that same peptide sequence. So this is an example of an ortholog. An ortholog is the same gene appearing in multiple species. We, for example, share genes with grass. Right? The grass growing outside, well, not so much in this heat, shares genes with us, right? We both have pyruvate kinase genes, for example, because we both do carbon metabolism. All right, so orthology is a common reason that a multi-species database will have a very complex time discerning which proteins are there. And people who are working in multi-species data sets, stuff like buckets of seawater grabbed out of the ocean, you're going to have lots of species there, and untangling which species you need to make a complete explanation for what you saw is hard. Also, a lot of genomes have grown in size through duplication. Does anyone work in trees? Okay, good. So that's a tough place to work because trees have some of the biggest genomes on Earth. When you compare the human genome size to one of those, I mean, we're small potatoes, guys. Those guys have lots and lots of copies of their chromosomes, and so finding the same gene in very slightly different sequences in the same organism is to be expected. But even the human proteome has a lot of paralogs within it, where we had a gene that duplicated itself back in our evolutionary history, and now those two copies have drifted apart from each other in sequence to take on different roles. So gene duplication may make it possible for the same peptide to be explained by either gene duplicate. And finally, lots of sequence databases report isoforms as different protein entries. So a gene that can produce 5 different isoforms will have 5 independent sequences that all appear in the database. And for the purposes of protein inference, that's very confusing, because which isoform is the best explanation for the peptides you've seen? Difficult, very difficult to solve. Does anyone here have an older brother or sister? Okay, sir, I'm going to ask you, did your older brother or sister ever call you a name? Did they call you something other than the name your mom gave you? Yes. Do you want to share any of them, or are they all? We don't have to go there. That's okay. It's okay. I have an older brother, and sometimes he watches these YouTube lectures, so hi Tom. School teacher in Missouri, look him up. Anyway, so my brother was very fond, when we would go wandering around in Walmart, of calling me cheap. He'd call me cheap. We'd be looking around in the toy section, you know, little Dave. I remember I had this Saturn V rocket in plastic, and I was like, I want this rocket, this is freaking awesome. And he would say, why don't you just buy it? I'm like, it's expensive, Tom. It's like, you're cheap. Right. Many years of this. If you, as a scientist, are called the equivalent, it's actually high praise. And that term is parsimonious. Parsimonious. In science, we should never reach a conclusion that goes beyond what our data can support. We should always be looking for the smallest conclusion that explains the evidence we've collected. So it's possible you can spin a brilliant story out of your data about this data set is evidence that Martians created all life on Earth. Okay, maybe that's a science topic, but it's not a scientific claim. It goes beyond what your data can support. Parsimony means that you're being economical in the explanation you're offering for what you've observed. It comes from William of Ockham in the 14th century, who said something along the lines of pluralitas non exponenda sine necessitate. Right? You don't want to be more expansive than necessary in explaining what you've seen. All right. So how does that work? There are lots of ways that this untangles. I'm going to be relatively brief about this. But I want you to imagine that the evidence we've accumulated through tandem mass spectrometry has a big list of peptides. Oh, did I make no sense there? Did you have a question? Can I help clarify that? Oh, okay, cool, all right. I thought I might have just jumped right past everybody, so feel free to stop me. All right. So imagine then that these yellow blurbs represent bits of peptide information that we've identified. Now, they're not all equal, right? So we have this second bubble up here that represents 10 different peptides that we identified. But we've simplified the graph. We've done so by grouping together peptides that match to exactly the same sets of proteins. Now, has everyone heard of xenografts? Mouse xenografts? Yes and no? A few yeses, a few no's. Okay. If you have a new cancer drug, do you immediately lurch into patients and say, here, take this, let's see what happens? No, of course not. Right. So, very good reasons why we don't do that. Instead, one of the things that we found we can do is to take cells in a biopsy from a tumor, inject them into a mouse, and see that that mouse now grows a human tumor inside it. Weird, but true, this works. So you can have human tissue growing inside a mouse. It's vascularized by the mouse's blood system, so it has mouse blood flowing through it, feeding it, but it's letting free human tumor cells that can go invading other parts of the mouse and stuff. It's a challenging area of research, but it gives us the ability to do our work directly on a human tumor grown in a host that doesn't complain as much. It's considered more ethical to do this to mice than humans. It gives us the opportunity to do research that we could not otherwise do, work that is quite necessary to extending human life. That's that only justification I'm going to put on it right now. Personally, when I was in cellular physiology class as an undergrad, I made an agreement with the other students that they would handle the mice and I would handle the spreadsheets. It was a much better division of labor. All right, anyway. So, I'm sorry, I've just outed myself as a total coward. Blood, oh! All right, so we have these peptides. At the left, we have proteins. Some of these proteins have been marked as human sequences. Some of them have been marked as mouse sequences. So because this sample contains both human and mouse proteins, we must include them both in the sequence database. And now we've got this complex, if you'll pardon the expression, rat's nest of associations between proteins and peptides. We need to figure out which of these proteins we're going to claim to explain the observed peptides. That's a mess. So the In the old 1990s days, things were really hairy, and it was very much a Wild West atmosphere in proteomics. Everybody wanted to publish the biggest inventory of proteins. Our technologies let us measure 80% of the known yeast genome. Our technology has seen a peptide from every protein in the yeast genome. Yes, if you allow your false discovery rate to sort of 50%, sure. Right. Lots of lies, frankly, floating around in the 1990s. So much so that the journals put together publication reforms that required people to meet certain criteria in order for their data to be published at all. And this did quite a lot for enforcing better behavior in a very misbehaving community. So, the rule here should be that if you've explained a peptide, you no longer need to re-explain it with another protein. I'm going to show you something kind of magical here. If you look at these two blue boxes at the left, we've drawn a double bound around them, right? So table 1X iso-A human and table 1XR1 human have double bounds around them. If you were to accept that these two proteins are present, all of those peptides at the right have been accounted for. So, parsimoniously, in proteomics, we would say these two proteins are sufficient to account for all of the evidence at the right. None of these others can be claimed as identifications. They are artifacts of the appearance of these of these two proteins having been present. So you can see that this cuts down the protein numbers quite a lot in these multi-species databases in particular. But I'm going to introduce another kind of complex frustration about this. In this case, did we identify no mouse proteins at all in this particular graph? Because you can see that the explanation we're reaching includes just these two human proteins, and this possible mouse protein fell away. That potential mouse protein fell away. In this case, I want to remind you that proteomics can provide you evidence that this protein is definitely here, but we have a substantial false negative problem as well. There may be other proteins present in the sample, and its absence from a protein list does not mean, in any case, that we've proven it's not there. Okay? Point of confusion, but it's true. We can tell you things that we know are there, but there are things there that our technologies are not sensitive enough to have detected. Okay, so in this case, you've seen a complex example get decomposed down to 2 proteins rather than 7 to explain all of these peptides. Any questions? Okay. Parsimony, using these principles, has a really dramatic effect on how many proteins we claim. In the case at the left, we were using a sigma protein standard that contained 49 human proteins. We know it contains exactly 49 because they purified each of those and then created this commish of the 49. We used 3 different database sources for this. IPI was a really old approach that was kind of a HUPO, EBI effort to say, what are the proteins we really trust for humans? It was pretty big. The Swiss Prot database was much, much bigger because it contained lots of different species. And the Swiss Prot human set was much smaller by comparison. In this case, we're looking at what happens if you say any identified peptide matching any protein in the database is justification for claiming that protein is there. Right? This is kind of like the bad old 90s definition of how to claim proteins are there. If any peptide you observe could match to any protein in the database, claim it. Name it and claim it. Terrible strategy. So here we have the maximal list. That's obviously way off the hook. For a sample that we knew contained 49 proteins, we would have claimed 400 in the Swiss Prot search. That's why a lot of garbage science happened back then. Grouping indiscernibles is an approach that says these sets of proteins explain these peptides. Any one protein from this set would be an equally good explanation for why we saw these peptides. So we call this grouping indiscernible proteins. Right? So just taking that one step can cut down those numbers quite dramatically. And finally, if you're applying parsimony plus this grouping of indiscernibles, we find that in all three databases, the number of proteins that we claim is much more in keeping with what we know to be present in those samples. The same is true when you work with samples in the real world. So these are serum proteins that have been run over a multiple affinity removal system to remove the most abundant proteins in serum. You can see that the numbers from Swiss Prothuman decline just a little bit because that wasn't a very redundant set. But again, when we look at the multi-species database search result, the number of proteins we claim was far more conservative after grouping together indiscernibles and stripping away the redundant protein IDs. So proteomic identification depends upon a database of known protein sequences to define the set of possible peptides that can be compared to tandem mass spectra. Controlling the error in identified peptides is critical to reliable proteomics interpretation. As somebody who's written a few systems for filtering PSMs and coming to careful conclusions about error rates and so on, I've had several people approach me to say, your software throws away my peptide. And when we look at the evidence for his peptide, we see it's trash. So lots of people fall in love, right? They have some result in their search results. They've got hundreds of proteins and they really love the story, their biological story they're going to tell about this one protein. And they feel sad when curation eliminates the evidence for their pet protein. We as scientists have got to be careful about falling in love with the story we're going to tell rather than what the data have to tell us. It's part of the job, unfortunately. Detecting indiscernible proteins and applying parsimony is a conservative guide to estimating how many proteins you've seen in a sample. There are lots of people who would like to escape those rules even today in proteomics publication, but generally speaking, peer reviewers and journal editors have helped align on this one, that people really do need to have sufficient evidence before making claim about identifying a protein in a sample.